Thanks so much for the opportunity to present my group's work. And in particular, I want to thank Sean, former student in my lab who did all of the work that I'll be talking about. Unfortunately, he was unable to be here, so you're stuck with me. This is function COSY. So what do we want to do? Predict functions of proteins, biological processes they're involved in, such as Go terms and the like. And proteins interact with each other to carry out cellular functions. So one popular way to predict protein function is from a protein-protein interaction network of a given species. In these networks, nodes are proteins and edges are interactions between them. Typically, this means physical bindings. And this applied task of protein function prediction can be translated to the computational task of node label prediction. Given a single network, objects of interest are nodes or proteins, and the goal is to predict labels or functions of those objects. Clearly, there are many other ways to predict function of a protein, for example, by studying the protein 3D structure, which directly dictates which other proteins a protein can interact with. And in this context, I argue, and I hope to convince you within the next 30 seconds, that a useful way to analyze 3D structure of protein, rather than doing so directly, is to first model the structure of protein as a protein structure network in which nodes are now amino acid residues and two nodes are linked by an edge if the corresponding amino acid residues are close enough in the 3D fold. And if you accept that, then I'm going to tell you a little bit more why we need to model structures as networks shortly. Then the computational task is now given multiple networks corresponding to multiple proteins. Objects are networks, and the goal is to predict labels or functions of those objects. And just tiny little flavor why study protein 3D structures as PSNs. Some of you in the audience have done so, Peja, for sure. My PhD advisor, Natasha Perjun. Sorry, I didn't mean to reveal that yet. There's a paper by Vladimir Grigorievich on structure-based protein function prediction using graph convolutional networks, which means take 3D structures, mold them as graphs, apply deep learning, and it happens to outperform in the context of protein function prediction, traditional sequence and 3D structural approaches, including top performance from the Kappa challenge. And my lab has done numerous studies in the context of task of protein structural classification, predicting cat and SCOP labels of protein structures using PSNs, and we have also shown superiority of modeling structures as networks first and then doing this. If you're still not convinced, we can chat later, but humor me and say PSNs are the way to analyze 3D structures for a moment. So now we merge these two ideas. This is the novelty of the study I'm talking about today. We have protein-protein interaction network of a species in which nodes are proteins, which themselves are networks at this lower scale period. And in general, and then the goal is to predict either function of nodes, labels of nodes in the network at this higher level, or labels of graphs at this lower level, but at both levels, objects are proteins. So we formulate this idea of a network of networks where nodes at level 2, that's the general terminology we use, level 2, are networks themselves, and this applies to any type of real-world data. If we had it, this could be social network of interactions between people, and if we would have protein-protein interaction network of each individual, how awesome would that be? Okay? So what we are asking is, rather than traditional level 2 node label prediction alone, or level 1 graph label prediction alone, what happens if we do network of networks based entity label prediction? Will we predict labels more correctly? If you leave with this, I'm happy. Feel free to zone out, check your email, fall asleep now. Now I'm just going to try to give you a little bit more idea of how we do this and what key results are. And again, it's going to be tiny little scratch. I'll point you to the paper shortly so you can read more details there or talk to me after the talk. Just to comment on novelty of this idea, the idea of networks has not been used in the context of entity label prediction and thus not in the context of protein function prediction. So we had to start from scratch, completely develop entire framework for how do we fairly compare tasks of NOM-based entity label prediction versus node label prediction alone versus graph label prediction alone. And there are a handful, 3 or 4 studies that do use networks of networks as we define them. A network whose nodes are networks themselves, but such studies differ drastically in several aspects from what we do, and we couldn't find a way to compare anything they do to what we do here. We couldn't use their methodologies in our task. And then there's a dozen plus papers that use the term network of networks, but they don't do this model I just mentioned. They instead deal with multiplex networks where you have nodes of different types at the same scale, or interdependent networks, or they deal with hypergraphs or alike, so they're not true network of networks as we define them. And I'm going to quickly run you through this just to give you an idea of what our methodology looks like. We developed the first ever synthetic model generator for networks of networks with different topological configurations that you can play with. Labels are picked up based on the network structure, so only methods that pick up structure correctly at both levels combined will be able to predict labels correctly. If our methods that we propose didn't perform well here, that would be horrible. The fact that they do perform here, it's sanity check that we do what we're supposed to do, okay? We also provide a biological network of networks with human protein-protein interaction network at level 2 and the PSNs at level 1. I think this is one of the key contributions of our study for now, because it's a proof of concept study, so feel free to take the data and play with it, do whatever you want with it. And I provide some statistics of how large the data is just for your information. And here the proteins are labeled by Go terms, and we focus on biological process Go terms, experimentally obtained only annotation data. And we study what we consider some of the state-of-the-art approaches on node level, graph level, meaning level 2, level 1, and they come from both graph theoretic and machine learning domains. We wanted to see how those 2 domains compare to each other. And then at the NON level, we kind of simply integrate information from different levels as the first proof of concept step, but also propose our graph convolutional network approach. Again, basic step, and that doesn't always work the best, and as I'm going to maybe illustrate on the next slides, but it was just the initial idea. Tons of room for improvement, no questions there. And we use some of the established ways to evaluate accuracy of these approaches at the 3 levels, meaning level 2, level 1, and the combined, fairly comparing things against each other. We have 30 different synthetic network of network configurations that we produced. And just a key summary statistic that at least one of the NON approaches that we use beats all node level and graph level approaches, which are single level approaches. It is never the case that single level beats NON. Again, this is just verification that these integrated approaches do what they're supposed to do, but doesn't necessarily mean anything spectacular. I'm going to skip this in the interest of time. Just to give you an idea, this is the best NON approaches. This is the best level 2 approach. So just to give you an idea of the level of improvement. For biological data, for real data, we study 131 Go terms after lots of data filtering. And we basically, for each of those Go terms, try to predict whether a given set of proteins has or doesn't have that Go term. So we basically report results per Go term. And of all the 131 Go terms, for 69 of them, or 53%, at least one of the NON approaches beats all single level approaches. So it's not always the case that NON wins, and that's fine. Ignore the figures that I'm about to show. I need to get to the second part of the table. But for 38 of these 69 Go terms, which is 29% of all Go terms, only network of network approaches produce non-random results, which means current approaches as done in the literature would completely fail for them. And that's the value that I think network of networks brings. It can help predicting functions that we can not predict with the current approaches, at least not in the evaluation framework we've used. And that hopefully justifies further studies of the network of networks idea. In terms of running time, network of networks approaches are slower by design, but still very feasible on our data. And we believe that if they can lead to new knowledge, then the extra computational complexity is justified. But hopefully we will not only yield in the future newer NON approaches, but also more computationally efficient. And again, if you take this away with you, if you deal with data that can be modeled as a network of networks, or you're interested to develop approaches for this purpose, look at our paper. I'll stop here. And again, thank Sean and the funding sources. Thank you so much.